It was the early 1950s, a time of post-war recovery, booming science, and unanswered mysteries. One of the greatest questions in biology still loomed over every laboratory. What is the structure of DNA, the molecule of life? Every living organism, plants, animals, bacteria, humans, carried DNA, but no one knew how it looked or how it worked. That is, until two ambitious scientists, James Watson, a young American biologist, and Francis Crick, a brilliant but unconventional British physicist, stepped onto the stage at Cambridge University in England. Test tube the race to understand life. Watson was just 23, energetic and bold, while Crick was in his 30s, loud and full of wild theories. Together, they made a strange pair, but they had one thing in common. They were obsessed with DNA. Other scientists, like Rosalind Franklin and Morris Wilkins, were also racing to crack the mystery using X-ray crystallography. Rosalind Franklin, working at King's College in London, had taken one of the clearest photos of DNA ever seen, a shadowy X pattern called Photo 51. That image was the key, though she didn't know it yet. Watson and Crick, meanwhile, weren't doing experiments, they were building models. They believed that if they could visualize the shape of DNA, they could understand how it functioned. DNA, the double helix revelation. In 1953, using information, some controversially shared from Franklin's photo, Watson and Crick suddenly had a breakthrough. They built a twisted ladder-shaped model, two strands winding around each other like a spiral staircase, with pairs of molecules, adenine with thymine, guanine with cytosine, forming the rungs. It was elegant, it was beautiful, it made perfect sense. This structure explained how DNA could copy itself, how it carried genetic information, and how it could mutate, which was the foundation of evolution. They had discovered the double helix, a symbol of life itself. Watson and Crick rushed to publish their findings in Nature Journal in April 1953, with a humble sentence that echoed across science. We wish to suggest a structure for the salt of deoxyribose nucleic acid, DNA. This structure has novel features which are of considerable biological interest. That was an understatement. It was one of the most important discoveries in history. Globe showing Europe-Africa, the legacy of the double helix. Their discovery opened the floodgates of modern biology. Suddenly, scientists could study genes, trace ancestry, cure diseases, and even alter life itself through genetic